Now, to temper expectations, so the brainstem is a very small structure, and we're slicing it into even smaller slices. This is the midbrain. In the anatomical position, so we flip it. So here is another good resource from Ed Lowe et al., the 100 micron resolution MRI, and that's the midbrain in great detail. On the other hand, we have here a cranial CT scan where it's not so detailed, but the overall shape is identifiable, like a square pants or a squarish looking Mickey Mouse. So this is a cut to the junction, the pontomesencephalic junction, where the midbrain transitions into the pons. So this is how it appears on the MRI, the high resolution MRI. At the pontomesencephalic junction, you start to see the bulge of the bridge of the pons, the belly of the pons, or the basis pontis. So this is the upper pons, where a majority are formed by the massive pontocerebellar fibers interspersed with the descending corticospinal tract fibers, which appear darker on the cross section. These are separated from the tegmentum of the pons by the medial lemniscus. And there, at the dorsal pons, at the fourth ventricular floor, you can see the two dark spots, which correspond to the locus coeruleus, which produces norepinephrine. Aside from the pontocerebellar fibers and the corticospinal tract fibers, the trigeminal nerve enters or exits the pons at this level. You can also see the brachium conjunctivum or the superior cerebellar fibers exiting the pons to go up into the midbrain. As you reach the pontomedullary junction, the pontocerebellar fibers become less and less, and the descending corticospinal tract fibers begin to coalesce, thus the change in the shape of the pons at the caudal pons. The brainstem slowly tapers down into a more narrow column until it becomes the spinal cord once it goes beyond the foramen magnum. This is the rostral medulla. It can be differentiated from the closed medulla or the caudal medulla because you can see there dorsally the tail end of the fourth ventricular floor, also known as the obex, under which we can find the so-called area postrema, the vomiting center. A continuation of the corticospinal tract at the level of the medulla oblongata is the pyramids, or just above the eventual decussation or crossing of the corticospinal tract at the cervicomedullary junction. Lateral to the pyramids are the so-called inferior olivary nuclei, or better known as the olives, a part of the cerebellar pathway. Meanwhile, the dorsolateral medulla contains mostly ascending sensory fibers, apart from the descending fibers for the face coming from the spinal nucleus and the tract of the trigeminal nerve, which will descend up to the level of, down to the level of the second cervical cord segment, or C2. Meanwhile, the caudal medulla is also known as the closed medulla because the fourth ventricle is no longer present. It begins from the obex until the medulla reaches the foramen magnum. It also contains the pyramids, the inferior olivary nuclei, as well as the dorsolateral area where the sensory fibers are ascending or descending as far as the spinal nucleus and tract is concerned. Of course, we must always talk about the cranial nerves when we talk about the brainstem. But when it comes to imaging, only the Fiesta sequence of the MRI will be able to visualize the cranial nerves very well due to their size. On CT scan or the normal or the usual MRI sequences, it is usually the trigeminal nerve that is best seen due to its large size. The remainder of the nerves may not be easily visualized. The cranial nerves become more relevant clinically or in case of neoplasms when there are nerve sheath tumors or schwannomas involving the meninges or the Schwann cell nerve sheaths that surround the cranial nerves. They may also be compressed by tumors in the area at the base of the skull or by meningiomas.